Thank you. It's actually, I don't know about you guys, but I'm having a lot of fun. Can everyone hear? How's that? Is that better? All right. I promise to be loud as I get going. So yes, I don't know what I am. If I'm not a hobbit, perhaps I'm a dwarf. I don't know, but we're wresting it back from the elves for now, and going to give you something that's very data heavy. And so I cannot promise to give you information as useful as the last talk, but I do promise that my talk will be shorter. And so I'm going to give you my plan to get things started. I'm going to focus on taste responses because that's what I study, but I'm going to try to explain to you why the stuff that I study is interesting in a more general sense, and why it should be interesting to physicists who are interested in approaching the biological systems. I'm going to tell you that taste responses are dynamic, and by that I want you to know that I do not mean that taste uses temporal coding. I mean that through looking at the dynamics you can see neural process, which is something very different. And then I'm going to go on and tell you that these dynamics imply and reflect interregional interactions. And you've heard a lot about this already. You're going to hear a lot more about it after. I'm going to talk to you about it in a little different way. But these dynamics are part and parcel of one brain area influencing another. I'm going to move on to talk about real-time population dynamics, so neuron-to-neuron dynamics. And I'm going to, at that point, get particularly nasty and tell you that classical analyses that are used in most studies of decision-making, giving you only a dirty window into those real-time population dynamics, and that decision-making does not work the way you have been taught, even in the best circumstance, or it may not. And then I'm going to bring this all together to show you how what this implies is that sensory cortex, at least in the case of our system, is actively involved, directly involved in the driving of behavior, which means that sensory cortex is not just there representing the stimulus for use by the motor system, it is actually the first part of the motor system. And so if I get that far at the end, because I know that my friend Siddhartha likes this sort of thing, I might throw in a little philosophy, but on the other hand, I might take pity on you. And so let's get started by talking about what people refer to as temporal coding. And while this is probably for this audience a no-brainer, it's worth thinking about the fact that under most circumstances, people do not think of time when they talk about what's going on in the brain. So starting with taste, according to many in the field to this day still, taste coding works according to what are called labeled lines, which is to say that in the brain, there are neurons that respond to one taste or another taste, and the way that you know that a particular taste is on the tongue is because the right neurons light up. Here is work from Chen et al. In 2011. This is work in which they did calcium imaging of the surface of gustatory cortex, which is what I'm going to be talking to you about today. And here you can see one area of gustatory cortex where a bunch of the neurons lit up when sweet was on the tongue, so when sucrose was on the tongue. And you'll see that these neurons did not light up when either of these other tastes were on the tongue. In fact, no other taste activated these neurons. Sweet did, according to these analyses. I'm actually not going to get into this in class, but I might suggest as an exercise, you try to figure out how these horribly wrong results could have been produced. And then the next question is, how did it get into science? So here is a different set of neurons from the same brain region, and you can see that these neurons, many of them were activated by sodium chloride, salty, and not by sweet or bitter. The conclusion of this study and of much work in the taste system has been that neurons in the area of gustatory cortex respond to one taste or the other. And you know that sweet is on the tongue because the sweet neurons light up. This study went one further and said that there are little patches that they called hotspots that were all of the activity related to tastes. I'm going to come back to taste, but I want you to know that this does not make taste, the taste field is not retarded. In olfaction, they recently gave a Nobel Prize to Linda Buck and Richard Axel for showing something quite lovely. This is actually a mouse head that has been sliced in half, And so what you can see here is the olfactory epithelium, which is the inside of the nose. And here is the cribriform plate, which is the bone that those neurons go through on their way to the first processing station of the olfactory system, the olfactory bulb. And what they've done, and what got them the Nobel Prize, is they managed to knock in a tracer that was hooked to cells that were expressing one particular receptor for an odorant. And so all of the neurons, all the olfactory receptor neurons that had that one particular receptor lit up, and what you can see is that they were spread all the way across the nose, but that when you got to the brain, they all converged on a single spot, or actually a few spots. So this is a schematic of it showing that you could have receptors all the way along here, but that all the receptors that were blue come into the blue spot. And that means that when you look at the brain and again do calcium imaging down at that first layer and you put on a particular odor, you see a particular set of spots light up. And the theory behind it, and I should be completely honest and say that this is the work of Tom Baza and Matt Wakowiak, who don't believe any of the bull that I'm slinging here. But the general belief is that you know what odor is on the nose simply by the spatial pattern of the neurons that light up. And thus, those neurons can be thought of as just either turning on or not turning on, or maybe turning on to different degrees. But there's no time here. Taking it one step further to something you're all familiar with, visual cortex. We've already seen figures related to this showing that in V1 you get neurons that respond to bars moving in particular directions. The typical analysis of this has the neural response on the y-axis, the orientation of the stimulus on the x-axis, and these beautiful, beautiful tuning curves that describe really very few of the neurons, but what the neurons are doing. But keep in mind that this is suggesting that what visual cortex does with the neurons is adequately described in a single number. The stimulus comes on, that's this orientation, and that's the number of spikes that describes the response to it. And so this is widespread. We still are in a world where, in general, you will deliver a stimulus, take a bunch of time after that stimulus is delivered, average across it, or take the peak instantaneous firing rate across it, and say that is what these neurons do to this taste. Now, to anyone who studies networks, you just have to think a moment to realize how silly this is. And so we're going to go back to taste now and tell you a little bit about the taste anatomy. One of the reasons I've gotten into taste is because I had the shit scared out of me when I saw the Van Essen picture that was put up for you a couple of days ago with all of those visual areas all over the place. Taste is a lot simpler, but it's still not simple. You have a tongue. Information comes from the tongue to the nucleus, the solitary tract in your medulla. In case you're interested, there are 3 separate cranial nerves that carry that information, because while nobody studies taste, taste is one of your most important is perhaps your most important sense for survival. That information goes to another brainstem relay in the pons, the parabrachial nuclei. You're not going to be tested on this. And then things get crazy, because from these parabrachial nuclei, neurons, at least 3 separate pathways, more like 4 or 5, in parallel lead that information up to the forebrain. Here we have 3 forebrain structures that take that input: the amygdala, a part of the thalamus called the VPMPC, and the hypothalamus. There's also the BNST and a few other regions. Some of these projections are from the same neurons in the PBN, where the axons branch and go to multiple places. And so now things are getting nuts, but then things get worse, because you get the canonical thalamocortical pathway that is in every sensory system. But in addition, the amygdala, and although I haven't drawn it, the hypothalamus also project to cortex. And that projection is reciprocal. And so now, if you're a neuron up here in cortex, you're getting perturbation from the taste is being felt by you via this pathway, and then via this pathway, which is a little slower. You're getting these things asynchronously converging, to use Miguel Nicolelis' favorite term, and then feedback to the brainstem. In fact, these brainstem nuclei, like so many other early relays in different sensory systems, 80% of the synapses down here are actually descending synapses from forebrain and not taking input from the tongue. And so now imagine you're a poor gustatory cortex, and I'll just call it GC, it's the primary taste cortex. You're a neuron up here. What's actually happening to you when a taste hits the tongue, and this is the simplest story I can give you, is that information comes up, wham, you get it. And then more information comes up, wham, you get it. In the meantime, you're sending that information back, and it's looping around. And so nobody but a hobbit would think that you could characterize the output, the response of this cortical neuron, or of any of these neurons, as we'll discuss, in terms of a single number and really understand what's going on. And so that's why we're going to talk about time. There's another reason, though, and this is actually the biggest reason I got into taste. And the reason is because there is tight perception-action coupling in taste. Assuming that you are working with an awake animal, and for the life of me, can't understand why you wouldn't, because a non-awake animal is not doing any of the things that the brain was evolved to do. So if you take an awake animal and you present it with a visual stimulus, you can probably work it out so that the animal has no response to the visual stimulus. If you're doing an auditory study, you can play the animal a tone, boop, and see no response to that tone because as far as that rat or monkey is concerned, that tone is meaningless. It is from outside his experience. You put a taste on an animal's tongue and you get a reaction. Because that stimulus is in the mouth, about to go to the stomach. It is absolutely important that the animal figure out what to do, decides what to do with that stimulus, and act upon it. So, for instance, if it's something that tastes good, like, for instance, sucrose, you will get a certain set of reactions that are reflexive, essentially, to that stimulus, and here is what they look like. This is a rodent that's in restraint, although I won't be talking to you about restrained animals during the talk. This is work of my first graduate student, Steve Grossman. You can see the forepaws of the little beast, and you can see her snout, and what you'll be able to see when the movie starts moving is the jaw itself. What you can't see is that Steve has just put an eyedropper full of sucrose into the animal's mouth, and it is going to make what are called lateral tongue protrusion, which is our fancy word for licks, as it gathers the taste up and brings it down its throat. And there it goes. That is the rat yum face. This is a rat that's very happy about what's in its mouth. It has made the decision to swallow it. It's gathering it in, and it does that whenever you put something that's tasty on its tongue, or whenever it gets it for itself. Contrast that to what happens if something tastes bad. When something tastes bad, you get an entirely different set of behaviors, the most common of which is called a gape. A gape is a big yawning face. I'll show it to you in just a second. The teeth protrude. What you can't see in this video is that the tongue is rolling forward in an attempt to eject the fluid from the mouth. In this case, Steve has used quinine, which is a very bitter taste, although it is delicious with gin. And you can see that the rat gapes in response. There's one. There's another. Here are little limb flails. This is the rat yuck face. This rat doesn't want what's in its mouth. It is going to make this behavior. And so when you look at neural activity, you are going to want to be looking for something that doesn't just represent the taste, but actually reflects the process of the animal making the inevitable decision to reject or accept the fluid. And just in case you thought this was just rats, here is a human infant who has just put a piece of sour lemon in his mouth. Human infants gape in response to these stimuli. These reflexes come back with dementia. It is only with socialization, where we realize that it's not polite to go at the dinner table, that we stop. In fact, the only difference that I can find between humans and rats in this regard is that this human actually, after he was done with this, put the lemon back in his mouth. And very distant, or relatively distant on the phylogenetic tree, I'm going to show you now some toads. These are toads from the Kalahari Desert. You think of toads as being a little innocuous beast that sits in your hand, and the worst it might do to you is pee. In the Kalahari Desert, toads are rapacious hunters. And so this toad is the scourge of any small beetle, which is a little ball of fat as far as the toad is concerned, and will just eat tons of them a day. And so we're going to see a little beetle that has evolved a wonderful defense mechanism. He tastes like crap. And let's see what happens when this toad attempts to eat him. Goes into the mouth, and that is a gape, made by an animal that really has very little brain to begin with. And just to prove to you that toads are not very smart, there's one, there's another, and our hero escapes. The moral of the story is these are behaviors that are part and parcel of receiving the sensory stimulus, and figuring out what to do with it. Cool thing for me, the lucky thing for me, is that these behaviors are long latency. While they are easy decisions that the animal makes all the time and doesn't get it wrong very often, you don't often swallow something and say, hey, I just swallowed that even though I hated it. They're still long latency. Using the stimuli that we use, they are typically take a little over a second for the animal to get by making innocuous behaviors to making the right response. This is, in fact, an EMG signal, electromyography, from the muscle underneath the jaw. And the taste is put here, and what you can see here, these bursts of EMG activity are licks that are innocuous, that are the same to all tastes. The animal's just going. And then you get to this point, and you can see that these big bursts, which are noticeably different and which you can automatically categorize, they are gapes. And so these things come at very long latency. I should also add what's going to become important for our story. They're highly variable in latency. It's a beautiful rhythmic behavior, but some trials it'll start here, some trials it'll start here, some trials it'll start here. There is a wide variability from taste to taste how long it takes. Just as there's long variabilities in latency in all of the decisions we make. When you're watching TV and some actress comes on the screen, you may figure out who it is one time in 1 second, one time in a second and a half. It's a natural feature of biological systems. They are long latency. The neural responses during this period are similarly protracted. I don't know when I started, so I don't know. Oh my goodness. So these neural responses are appropriately protracted, which means that you can watch them across this entire interval, which means that taste provides an ideal medium with which to explore the intrinsic dynamics of a perceptual neural system, the construction of behaviorally relevant responses to meaningful stimuli, and that's what my entire talk is going to be about. I should mention in passing that I know that there are many kinds of temporal codes. I'm going to ignore most of them because I'm going to tell you about what we saw. Happy to talk to you about them later, why we don't see some of them, why you might, what the different ones might mean. But I will mostly leave that to my fellows to talk about. All right, and a moment of methods. We do the kind of technique that is now becoming quite common. Here is a coronal slice of the rodent brain. The rat brain is not so different from yours. It's just a little smaller. And here is where primary gustatory cortex hangs out. Almost everything I'm going to report to you is from bundles of electrodes dropped into this region and chronically recorded from, action potentials pulled off of these. I'm going to be talking to you about ensembles of neurons simultaneously recorded, but I should note that everything I'm going to talk to you about are small ensembles, because I come from a background in which I started my career doing recordings with sharp 5 megaohm impedance electrodes, recording from Purkinje cells in the cerebellum, where the action potential was like this, the noise was like this, and the way we handle my disappointment and disillusion from the low signal-to-noise ratio with the kind of techniques you can use with freely moving animals is that we have very conservative standards for what is and is not a single neuron. And so I'm going to be talking to you about ensembles that are only 6 to 12 neurons in size. And I want you to keep that in mind when you see what are the striking abilities to predict behavior that we can get on the basis of those. And we do that in a rat that is awake and moving around and receiving a naturalistic set of stimuli, but the stimuli are being delivered through an intraoral cannula so that we can get complete instant tongue coverage, which helps us interpret our recordings. And so with regard to a discussion that Siddhartha and I were having, I like naturalistic and reduced preparations, and I try to do them both at the same time. And so what I'll do now is something that's always a little depressing. I'm going to reduce my 5-year postdoc at Duke University to a single slide. Because there's a long way I want to go with you on this, I'm just going to tell you and ask you to buy the copious amounts of analysis with lots and lots of p-values that went into telling us that, in fact, there are 3 epochs of a cortical taste response. Here is a set of peristimulus time histograms. You've seen things like this before, but I'm just going to reintroduce you. Firing rate up here, time post-stimulus delivery here. These overlaid are the PSDHs to a set of different taste stimuli. I'll explain why we use these in just a couple of minutes. And you can immediately see that nobody in his right mind would attempt to say, oh, let's just average across this time and give a single number and think that that describes this response. But in fact, it's better than that, because the response breaks down into epochs that are actually understandable and characterizable. And so to give you backing, what you can see is that immediately after the stimulus hits the tongue, there is a burst of action potentials that is fired to all tastes. This is a transient response. It is completely non-taste specific. In looking at this record, as scientists, we can say, aha, a taste has hit the tongue. And so we refer to that as the detection epoch, because during this period, the firing just tells us, okay, there's something on the tongue. We can detect that. But then you can see that within a couple hundred milliseconds, the responses to different tastes diverge. Now, in this period, this neuron is firing more to one taste than another, and more to that taste than a couple of others. You get taste information from this neuron. And so we refer to that as the identification epoch, not because we can say for sure that this is what the rat is using to identify the taste, but because, I'll just walk around so I block different people at different times, but because we as experimenters can identify the taste from even a small ensemble of neurons looking at just that period of firing. And then you can see that the firing rates shift again in this period, and now we get a new taste-specific arrangement, but now it's better than taste-specific, because now every neuron, while there are a lot of different ways the neuron might fire taste-specific here, when you get out to here, every neuron is essentially doing one of two things. Either they are firing most to the worst, most aversive taste, quinine, and least to the most palatable taste, sucrose, or the inverse. At this point, the firing reflects the palatability of the taste and the decision that the animal is about to make, because this happens a little bit before the animal makes the behaviors. And I'm going to come back to this. This is where I'm going to end up, because I'm going to show you that this is all averages. I'm going to show you that this is true in single trials by the end of the talk. And so here you can see that we have what amounts to palatability decision-related information, and you can actually quantify this very simply. All we do is we do a correlation. We correlate the known palatabilities of each taste with the firing rate to each taste at in each of individual time bins. And when you do that, you can see that you get nothing in the way of a correlation between those two values until around or a little after half a second, even though during this period there is taste-specific firing. And then it begins to rise and comes to a peak around the time that the behavior is emitted. And one of the things, this was 2001, well, this is this is a neuron that was recorded by Brian Sadaka in my lab recently. This is this basic tripartite characterization was something we came up with at Duke in 2001. And at that point, we were super excited to see this, because this looks like the classic ramp. The classic ramp that you might see in a decision-making model where information is being integrated across time, and maybe there's a threshold here, and that's when the behavior is emitted. And I'm going to come back to that because it turns out that we were wrong. But this is what taste responses look like. And so you can see that it's a process. If you're thinking as a psychologist, you can imagine that you're in the head of this rat, and you like, at the earliest time point, there's something on my tongue. Hey, it's quinine. Ew, get it out. You can see that occurring through time. Okay, but that's all phenomenology. We have to, if you're going to actually say that this is what is going on, you have to test it. You have to perturb the system and characterize it. We've done that in any number of ways. I'm only going to tell you briefly about two, one of which involves changing the palatability of a particular taste. We use a paradigm called conditioned taste aversion to do that. And all of you who remember your college years will know what I'm talking about here. Conditioned taste aversion is a very simple paradigm in which you have a tasty experience with some beverage, and then later you have a less enjoyable experience with a toilet, and then the next day, when someone offers you the beverage that at this point you considered to be delicious, the taste and the smell will be aversive to you. 30 years of work by psychologists has shown that this changes the palatability of the taste without changing its identity. So sucrose is still easily identifiable as sucrose. It still has the same similarity structure to other sweets and other tastes, but now it tastes like crap. And so with this paradigm, we can predict that if the characterization I just showed you is correct, changing the palatability of a taste through conditioned taste aversion should only change the response in that third epoch. And in fact, that's what happens. So here is an example of two PSDHs. I've overlain the pre-CTA response to sucrose with the post-CTA response to sucrose. What you can see that for both of these neurons, there are responses early. In this case, it's inhibitory. In this case, it's excitatory. But those responses do not change with conditioning. But then you can see that afterwards, the responses are different. And in fact, one simple way to analyze this, which I'll talk about a little bit, is doing a rolling PCA, principal component analysis, and then you can put the responses in a space. What you can see is that the pre-training response and the post-training response parallel each other until about 800 milliseconds in, and then they diverge. And so by changing the palatability of the taste, you change the response in the palatability-related epoch. But I want to talk to you about a tougher test we did on this. And again, I should mention that we have about 6 papers that are testing this characterization, papers that adjust that challenge the identity epoch and not the palatability epoch, shift the identity epoch around. But the test that we've done most recently that I'm kind of pleased with involves a sodium chloride concentration range. Why is this a useful set of stimuli to use to test this? And this is again in the work of Brian Sadaka. Because if you look at the palatability of these tastes, of these sodium chloride concentrations, and in addition to using those mouth movements, you can also just give the animal brief access and ask how much it licks. What you find is that the animal licks a lot to sucrose and licks less to quinine. This line is how much it licks to water. And the cool thing about this concentration range of sodium chloride is as the concentration increases monotonically, and I'm only going to say monotonically because we're not exactly sure what's the right scale to use to see if it's linear. But while there's a monotonic change in concentration, there is an inverted U-shaped change in palatability. The lowest concentration tastes like water. At about normal saline, the rat loves it, and so do you. At higher concentrations, it's basically seawater, and the rat doesn't like it at all. And so when you take this concentration range and record the responses in gustatory cortex, and then I'm just going to use to do a simple population analysis, again, the principal component, the first principal component, what you see is this, that the responses beautifully follow the palatability, but only starting at about 850 milliseconds in. Before that, they follow the concentration. And so the responses to sodium chloride shift so that during the earlier part of the response, you get a monotonic increase in firing or decrease in firing with concentration. You get out to 850 milliseconds in, and you get this whopping correlation with palatability. So we're pretty confident that this theory, this characterization that we've developed, actually reflects the processing of these stimuli. At this point, I want to mention to you that similar phenomena have been observed in other systems. This is work from the insect olfactory system by Gilles Laurent and his student Ofer Mazer. And I don't really want to get deep into it because we got places to go, but you can see that these different neurons, these are principal neurons that they're recording from in the first relay, have multiphasic responses to different odors. They've looked at this a lot, but they haven't looked at it in behavior, so they haven't been able to characterize what the different phases might, what the content of the different phases might be, but they reliably see this sort of thing. And here is the way they do analyze it. This is actually similar to what I've shown you. They put the whole thing in principal component space, and they show that here's pre-stimulus firing, puts a population right here in the space, and then as the smell comes on, you get a trajectory through space of the response. That's this response changing through time until it comes to a resting point over here. Different odors have different trajectories, and so they say these are different temporal codes for the different odors. This is just showing you that it's replicable. I want to mention briefly, but this would be a good time to point out that, again, the way these responses have been described is that this temporal code defines the stimulus, and that as the stimulus trajectory goes through here, that trajectory is what the code is for that odor. That's preposterous because in olfaction, as in taste, the animal can actually identify these stimuli ridiculously quickly when it has to. Here is an example from, this is work from 1972. Back in 1972, you could get this kind of work into the journal Science. This is showing licking of the animal. Each one of these hashes is an animal licking. You notice that the licking is very rhythmic. Rats lick at about 6 to 7 hertz. And you can see the licking to water. Then you can see the licking to sucrose. This is actually, the reason that this is less than this is because the animal it was his first time exposed to sucrose. He was a little shy. And here, The animal has been getting a taste aversion to sodium chloride, and you can see him licking away to water. Sodium chloride comes. He finally takes a lick, and that one lick is enough. The animal realizes in approximately 250 milliseconds, okay, that's not something I want to continue with. That's something I do not like. If you actually had to wait through a time-extensive trajectory to identify a stimulus, you would not be able to perform. What's going on in these temporal codes is not a time-extensive code for the stimulus itself. It is the ongoing process of figuring out what to do with the activity that's coming into the system. And I should note that the very fastest taste decisions, or the very fastest taste behavioral responses, in the best of circumstances, can be made just after the identification epoch begins. But point out now, and this is controversial stuff, that you even see similar phenomena to this in the visual system. And I'll admit, I'm not a visual systems researcher. I don't want to be one. And one of the reasons is because so much of it is done with stimuli that are meaningless, and it's so hard to control it. But here is actually work from Sugasi et al. Back in '99, in which they were looking at fusiform face gyrus. So here is a group of people that are looking at stimuli that matter to a monkey, the faces of other monkeys. And they're actually deciding to analyze those responses, which are typically just analyzed in terms of a single number. Ooh, it's a face neuron, it responds to faces. And instead, they're analyzing it through time. And what this ridiculously complicated graph shows you is that when that face is shown, the information relating to whether or not it's a face comes on very quickly and peaks here. At that time, information about which face it is begins to emerge in a separate epoch and peaks here. And while they don't have this in this graph, what happens then is a third epoch of activity, which is related to the emotional properties of the face. Just as the third epoch in taste is whether it's yummy or yucky, this next epoch is what emotion is being delivered on the face of this other monkey. Why is that an emotionally important stimulus? Because if the monkey is looking at you like this, you'd better run. And so the same things can be seen in other systems when you look at them right. Okay, so that's the first part. Taste responses are dynamic. Now let's move on to the next part. These dynamics imply and reflect interregional interactions. This is a summary of about 6 studies that we've done, 7 studies, showing that if you go through time, here is gustatory cortex. We have these epochs that we laid out. I've put them as if there are severe dividing lines between the two of them. There actually are. Between the three of them, there actually are, but I haven't gotten to that yet. So far, we've been talking about ramps from one to the next. But what I can tell you is in the basolateral amygdala, you see coupled dynamics. In lateral hypothalamus, you see 2 types of neurons, both of which go through coupled dynamics. In central nucleus of the amygdala, with a bizarre thing happening here that we think we can explain, you see coupled dynamics. And even in the early relay in the pons, you see coupled dynamics. So this whole system, while we typically look at each brain area separately and talk about what it does, they're all working on the same dynamic clock. Furthermore, it turns out that BLA, basolateral amygdala, which is often described as being the seat of emotion in the brain, where the emotional properties of a stimulus are determined, and that information is then sent to cortex for the cortex to use it to drive behavior. When you knock out BLA, you have this huge impact on GC taste responses. Here's a set of peristimulus time histograms of a single neuron to a bunch of tastes with the BLA intact. And here is what happens when you knock, and so this is another, neuron that does not really respond to any of the tastes. And then we knock out BLA. In this case, we knocked it out pharmacologically using infusions of the GABA agonist mucimol. And what you can see is that for this neuron, knocking out BLA had a crushing impact on the taste responses. Not complete, and we're going to come back to that in just a second, but keep in mind that the thalamocortical pathway is intact. But knocking out this side loop has a huge impact on these responses and has a huge impact on these responses. In fact, when BLA knocks out, when basolateral amygdala is knocked out, suddenly taste responses appear where none were before. I should mention that this happens 3 times as often as this happens. But so these brain areas are interacting. Even though the thalamocortical pathway is intact, showing normal taste responses is dependent upon the patency of basolateral amygdala. And despite the huge impact BLA has on taste responses, they don't knock them out. And so this is something that we've seen in several forms already. We're going to see more of. This is basically a classification algorithm. I don't think I need to go into it. It's basically we're using jackknife cross-validation, looking at whether when you have a set of responses and then you have a new response, how does it get categorized? And what you can see is that with BLA intact, these neurons give you a pretty good idea of which taste is on the tongue. When the taste is sodium chloride, the algorithm guesses sodium chloride. This is what happens when you knock BLA out. You can still do it just fine. In fact, as far as we can tell, there's really no decrement. On the other hand, what you do lose, as expected, is palatability. This is palatability-related information ramping up in the intact animal. This is palatability information ramping up to a lower level in the animal without a BLA. In fact, if you just look at the neurons where the responses were inhibited by BLA, X, while they still have taste responses, they largely no longer have palatability-related responses. Palatability-related responses normally go up from the middle to the late epoch. They don't with BLAX. And so BLA is vitally important for what taste cortex is doing. And now, recovery is 5 hours after. We were actually kind of surprised at the fact that this recovery was not complete. When we go out to 24, it is. But moving forward, because this is all well and good, it's predicted by classic systems neuroscience theory. Now I want to show you something that's actually a little more surprising, because these brain areas are not just connected to each other. In the taste system, they're interconnected between modalities. And so I want to talk to you now briefly about olfaction, and I want to talk to you about another cute little behavioral paradigm called social transmission of food preference, which we did with rats, even though the artist drew this as a mouse. If you take a rodent and give it something to eat, and then that rodent has a little tete-a-tete with a second rodent, what will happen is this first rodent will breathe on this second rodent, and the second rodent will say, oh, Ralphie ate a banana and lived. And then later, when you give that second rat the choice between two flavors, he will choose the one that was demonstrated, the one that was on the breath of the other rat. And this is highly reliable. This happens, you do this with 10 pairs of rats, you get this 10 times. And it's, again, obviously of evolutionary value. That's the way one rat knows what it can and cannot eat. And I should mention that this happens even if the taste that we're talking about is nasty. We actually in our lab sometimes use raw cocoa. If those of you who think you like chocolate have ever made the mistake of putting baker's chocolate or raw cocoa in your mouth, it's very, very bitter. Even in that case, you get this result. The second rat says, okay, yes, I know it's bitter, but he's okay, and so I'm going to eat it. So this is an olfactory task. In fact, you can knock out everything but olfaction during this interaction, and it will still work. The animal does this by the sense of smell, all of which makes it more surprising that taste cortex is necessary for this task. You cannot do this olfactory task without the taste system. Now, mind you, everyone should know that the taste system and smell system are necessarily linked. You eat what you smell what you eat. But still, this is suggesting that these systems are not separated. And furthermore, it works whether you knock out taste cortex at training or at testing. Either way, it works. And finally, the really most interesting part of this result is that if you inactivate taste cortex during both sessions, knock it out during training, and then knock it out again during testing, you rescue the learning. The interpretation that we put on this is that taste cortex is modulating the activity of the olfactory system, such that when it's knocked out, things don't smell right. But if you knock it out again, it doesn't smell right again in the same way, and the animal is able to recognize it. But of course, this requires testing. And so I should apologize. We're going to go briefly into a very different style of slide, because this is work that has not yet been published by my postdoc, Joost Meyer. And so he's much hipper than I am, so he uses black. And so he is looking at the fact that taste cortex, he calls it TC instead of GC, sends a projection to olfactory cortex, as does the olfactory bulb, and he was proposing that this pathway is responsible for what I just showed you. And so first thing he had to do was show that this pathway actually had an impact, and here is what you're looking at. Our PSDH is showing you taste responses in olfactory cortex. And furthermore, it turns out that these taste responses, they survive. They do not survive Anbesol on the tongue. Anbesol numbs the tongue. If any of you who have children, rely on Anbesol because it keeps your kids from screaming with toothaches. And so basically, this shows that the information in these PSDHs is coming from the tongue. Furthermore, when we knock out the nose, you can actually temporarily destroy the cilia in the nose that help you smell things. You actually completely don't lose any of these responses. So these responses are true taste responses coming from the tongue. And it turns out when you record from these two regions simultaneously, you can see that taste responses in taste cortex, almost always with one sole exception, lead taste responses in olfactory cortex. So it's clear that taste responses are going from taste cortex to olfactory cortex. To drive this home, we are now using optogenetics to demonstrate that without that pathway, you lose those taste responses. I'll come back to why optogenetics is like the biggest used car deal of the century in just a little while. But in the meantime, you can see that the taste responses in olfactory cortex are dependent upon this, and that in itself is still an okay, or it's not so surprising result, but you know what's coming. It turns out that when you knock out taste cortex, you change the responses in olfactory cortex to olfactory bulb input, to olfactory stimuli themselves. Here is an example of a response to an olfactory stimulus in olfactory cortex that is basically removed when taste cortex is inactivated. By the way, we used virally directed ARCH-T, which is an optogenetic silencer. But here in the same animal is another olfactory response that did not exist until you knocked out taste cortex. And so what you can see is while knocking out taste cortex knocks out taste responses, it modulates olfactory responses just as we would expect. And I should mention that while I don't have it here, we've done two more behavioral paradigms to show reliably that in fact the animal cannot do olfactory behavior tasks without taste cortex. And so yes, what this tells you is that these dynamics which are expected because the system is distributed and there's lots of reciprocal pathways and feedback and crosstalk, has surprising implications, which is to suggest that you really can't break down the system, or you shouldn't break down the system the way we classically do into separate modalities, and as I'm going to show later, into sensory versus motor. All right, and so I've gotten halfway through the talk in half the time. So now I want to talk to you about what this shows with regard to looking at real-time analysis, and why this shows where a lot of the things we think are true, or some of the main things we think are true, are not. I'm going to take you back to this picture, which I showed you at the beginning. Here is that gorgeous ramp, that gorgeous accumulation of information about palatability that results in the output. We loved it. It was great. And then we started looking at the damn data and realized that our data don't really look like this. These are PSDHs. In these, you lose real-time dynamics. Every one of these lines is a bunch of trials that you've delivered and averaged across or summed through. And when we look at the real data, instead of looking at the response of a single neuron across a bunch of trials, and instead look at the response of a simultaneously recorded set of 9 neurons in a single trial, you see something else. Each one of these hash marks is an action potential. This was a delivery of sucrose, not that it matters. And what you can see is something that really doesn't look gradual. And I want you to focus on these 3 neurons, and I'll give you a little line to help you out. To the left of this line, the first 800 milliseconds of this response, neuron 9 doesn't fire. Immediately after this line, it's firing a bunch. It's firing at a fairly high rate for taste responses. At around the same time, neurons 2 and 3 go from firing quite high to firing very low. One trial. Here's the very next trial of sucrose. It's this exact same phenomenon with one problem. You got to move the line. And so you can see the same progression of firing rates, sudden changes, but the change happens at a different time. Here's the very next trial. Once again, you see the same thing. I'm going to move the line for you helpfully. Low to high, high to low, suddenly. It doesn't look like there's a gradual ramping in firing rates going on here. Instead, what it looks like is that there are a pair of states, ensemble states, that we're going to now, to avoid any controversy, just say they are defined by the firing rates. And so there's a set of firing rates for the neurons in state 1, and then there's a set of firing rates for the neurons in state 2. There is a sudden transition from state to state. That transition happens at different times and different trials, which is what causes an artifactual ramp. We're going to come back to that. That's what this is all about. But what I've done here is I've given you raw data, and I do not believe in raw data. I think that, that journals that tell you need to show more raw data, reviewers that tell you that you need to show raw data, are reviewers that don't understand that the truth doesn't just stare you in the face. We have to do an analysis. And so we used an analysis that's very familiar to some of the people in the room, hidden Markov modeling. For those of you who are not familiar with it, it is the analysis that you face every time you talk into a phone to a machine. What the analysis is designed to do is to take a multivariate input and describe it as a set of states with transitions between the states, transition probabilities. And so in the case of a voice decoder, the input is the frequency ranges of your voice, and the states that it's attempting to pull out are syllables. And it can give you an output that can be looked at like this. It's not a PSDH, although it does have time on the x-axis, post-stimulus delivery. On the y-axis, what there is the probability. According to the analysis, it's how confident the analysis is that the set of neurons is in one of the states that it has uncovered, one of the sets of firing rate states. And so what we would hope to see, given what I've just shown you, and I'm just going to say right here, is that what happens is there's a high confidence that it's in a particular state, and that there's a sudden transition where that state becomes not likely and another state becomes highly likely. That's what we would be hoping for on the basis of what I've just shown you. The talk would be done if that's not what we found. Here is the real data, a single trial of sucrose. I've overlain the spiking itself from the set of neurons on the HMM solution, and what you can see is that multiple neurons change rates simultaneously or near simultaneously and precipitously at the boundary that is described by the HMM. I should mention, those of you who have seen ensemble recording talks before, like say hippocampus talks, are used to, whether you realize it or not, hearing about huge ensembles of neurons being recorded and maybe 5 to 10% of those neurons being involved in the actual process that's under discussion. 50% of the ensemble, a little bit more, change their firing rate at each identified transition, which is to say that every neuron is involved in this. This is not sparse. And by the way, we can putatively identify pyramidal cells and interneurons, and it makes no difference. Different cell types equally involved in this process. So here is an almost complete, and that there are minimal state-to-state transition times. Here is an almost complete data set. One of the only downsides to studying taste is because the animal will only drink so much so fast. You can only deliver a couple of trials per minute as opposed to the one every 3 seconds in vision, and or more. And furthermore, you are faced with an animal that is going to eventually become full. And so this animal was going to give up the game in about an hour. And so that is at least half of a complete data set. And what you can see is you get the same pattern for again and again for each taste. Furthermore, you get, although you really can't tell from here, different patterns to different tastes. And most notably, you can see that whereas in one trial, state 2 might end here, in the very next trial of the same taste, it might end there. And so at the very least, this suggests that when you collapse across trials in something like a PSDH and ignore the fact that neurons are working together, you are making some dangerous decisions. You're making decisions that deliberately circumvent things that are in the data. So I will step back for a moment and tell you that while I'm going to go on with the rest of the talk, talking to you about all of the great things about what we found, I want you to note that we're lacking something. This analysis makes an assumption that while there are dynamics, changes suddenly happening here, within a particular state, what happens is stochastic. Each neuron has a firing rate. It doesn't matter what the actual pattern of action potentials is. All that we're worried about is that the firing rate of each neuron, as if it's stable. We already know that that's not true. We haven't dealt with it yet. The analyses, the modeling becomes much more complicated and requires much better data and much more data to get away from this. But I just want to show you briefly that here is a single neuron's action potentials, and this is work by Sean Escola at Columbia, working with Liam Paninsky. And what we did was we did the HMM. So first, when you look at this neuron's firing, you see something that looks exactly like we expect. There's an initial, in this case, inhibitory response, second epoch, third epoch. But now, if you do HMM, identify what state the network was supposedly in according to the analysis, and realign your data according to the state transition, you now see that what's going on in this state is not stochastic. There is actually intrinsic dynamics within there. We don't deal with that at all yet. I just wanted to make sure you know, though, that we should be and we're not. But back to the good, or back to a different kind of bad. This trial-to-trial variability in when state transitions occur can explain ramps, can explain seeming accumulation as being an artifact of the analysis. Here is, again, this is also work of my then graduate student Bryant Sadaka, which is now in revision and hopefully safely passed the sharks. And here are a set of about 10 trials of a particular taste. And what's plotted here is the onset time, according to the HMM, of the state that was dominant around 1 second in. And I'll tell you why in just a second. And what you can see is that in each case, the HMM suggests that it happens suddenly, but that it happens at very different times from trial to trial. The important thing is that if you just average across these trials in terms of probability, you get a beautiful ramp. And it's a ramp that looks a lot like the ramp of palatability-related information. So the obvious possibility is that, in fact, palatability doesn't come on, the decision variable doesn't come on slowly and gradually. It, in fact, comes on suddenly, but at different times in each trial. That it comes on like this. The animal doesn't know what it thinks of the taste, and then it does. And so we decided to check that out. And so here is the original data, looked at a slightly different way. We call it the choice index. This is averaged across every neuron we recorded, including the ones that did not respond at all. And you can see that at about 0.5 seconds, palatability-related information rises and peaks at about a second, as I've been telling you. And then we did a very simple thing that I've already sort of presaged. We did hidden Markov modeling on these data. We made a simple assumption, which is that state is the palatability-related state, because it is the state that dominates at the time that we already know palatability-related information is high. And so we took that state and we said, when does it come on in each trial? And by the way, you can do this analysis using any number of criteria. When it crosses 0.5, when it crosses 0.8, when it asymptotes, it all comes out the same. And we then quite simply shifted the data slightly. These shifts were really small, on average about 150 milliseconds per trial. But now, instead of being linked to the onset time of the stimulus, it's all linked to the transition time into this state. Then we took these data in this form and just put them back through this analysis and got this. So you can see that when you do this, you expose a much more sudden transition into choice, that the decision variable comes on very suddenly. Although it still looks like it could be a ramp, and furthermore, you've already heard the dangers of significance testing. It's hard to say. I'm sure we could come up with analysis that says this difference is significant, but we'd like to do better than that. So what we did, in order to look at this and to answer the question, how steep is this increase, we actually ran a whole bunch of controls. I'm going to tell you about these controls, and normally at this point, I'm very, very, very specific and technical about what each control does. And I'm going to leave some information out, because your exercise for me today is going to be to explain why 3 different controls are needed instead of just one. So the first control analysis is a PSTH-based simulation, and this is because there is a big, nasty fact about HMM, which is that it's designed to do this. It will take crapola data and do its very best to identify states and transitions between states. And if your data has variability in it, it will hook itself to what looks the most like a sudden change in firing rates, in the case of the way we use it, and will identify that. And so it will improve what you see even if there are no sudden changes in reality, and even if there is no link coupling between neurons. It is inevitable that it will help. And so what we did was we did a PSDH-based simulation, where we took the real data and the PSDH for the real data, the bunch of trials to a single neuron, and then we used this PSDH to simulate a bunch of new trials. And so what you can see just by looking at it is that the PSDH of the simulation looks like the PSDH of the real data, because that's what it was built from. But that you can see that the simulated responses, essentially the difference is that each simulated response is a good reflection of the PSDH. Each one has a ramping up firing rate at about the same time, whereas in the real data, there were sudden transitions at different times on different trials. And so when we take these data and plug them back into our analysis, we are going to see just how much of the phenomenon I've showed you is a result of just the analysis itself as opposed to our data. Because these simulated data retain all of the temporal coding that could be seen in the PSDHs. All that they lack, almost all that they lack, is within-trial coupling between neurons and sudden transitions. But we did a second control analysis, which is called within-taste, within-neuron shuffling. You're probably all familiar with that, but I made this cute little diagram, so I'm going to show it to you. Let's say you have 4 neurons, you've delivered 3 trials of a taste, and you think that the system goes through a set of 3 states with different transition times and different trials. If you take trials 1, 2, and 3 and add them together, you get the PSDH. And it doesn't matter what order they're in. Addition, 1 plus 2 plus 3 is the same as 1 plus 3 plus 2. And so as long as all of those 3 trials are up there in that row, you're going to get the PSDHs. You're going to retain the PSDHs. And so what we did was we kept things that way, but we shuffled trials within rows. And so once again, you can see we've eliminated the within-trial coupling between these neurons' activity, but we've retained all of the information about the PSDHs. And so this is our second control. And so we did a third control which I'm going to tell you about in a minute. But in addition, we decided to model the perfect data, because HMM has a second problem, or the procedure has a second problem. While it is true that HMM will do its very best to produce states and sequences, the whole process also causes smearing. The reason is quite simple. Let's say you have a system that goes through 2 states. You can't see the states, though. That's why they're called hidden Markov modeling. All you see are the spikes that are output. In this situation, there is no way you can know where in that time period the transition actually occurred. The analysis is going to be confused. And in fact, it's actually worse than this. Since we're looking at firing rates, you need a finite amount of time to actually stabilize what the firing rate might be. And so there is going to be a period of time in which the analysis cannot be sure what's going on. And so this analysis, while it will sharpen things up, it will never sharpen things up all the way. And so what we decided to do was to look at the perfect step function data. We got the rates and transitions from the real data. So we took the real data, we did HMM, we got what the states were and when the transitions were, and we simulated perfect data using these precise rates and transition times. So this simulation, the data looked pretty much like our real data, the only difference being that it was perfect. All of the neurons transitioned at exactly the same time from one rate to another instantaneously. All right. So now let's get back to actually looking at what we found and answering the question, just how steep is the onset of palatability-related firing? I'm looking at suddenness here, which is, rather than slope, is not confounded by the height of the change. And so suddenness is just how much time it took to go from low to high. And when we used the PSDH control data, and we looked at all of the 100 simulated data sets we made, you can see that there was a distribution of suddennesses, and the real data is out there. So what this tells you is that never did the PSDH-based control data transition as suddenly as the real data, even when you put it through the HMM realignment redoing. And so, if you are interested in p-values, this 100 data sets, the real data is better than 100 data sets, so the p-value is p is less than 0.01. And I should say that for the shuffled control, looks pretty much like this. I don't have it in here simply because we just finished it. But it looks pretty much, it overlays this. It goes a little bit to this side. It's a little bit better than the PSDH-based control data. That reason is for you guys to figure out. But it still, it does not look like the taste data. We are far outside this distribution. And then we finally decided, though, that the real important thing was to come to a conclusion about which model best describes our data. Is it the step function control data? And so what we've got here is we've plotted the difference from the real data for 100 step function control data sets. And what you can see is that it's centered on 0. In fact, we could discern no important differences between the results using our data and the results using perfect data that changes instantaneously. As far as we can tell, our data go from no decision to decision like this. And then we did one last control, because everything I've told you about with the PSDH control data and the shuffle control data still isn't sufficient, because the model that is used to describe these ramps is the drift diffusion model, which, as you've already heard about a couple of times, each trial is modeled as a random walk, a biased random walk. And given that, these two controls don't actually cut it, because you're going to get sharper transitions from the DDM than you do from either of those other controls for reasons that you have to tell me. And so we modeled another simulation. We did another 100 simulations using the drift diffusion model directly, and this is the result for their difference from the control data. The answer is they look less like the real data than a step function does. So our data are more like the step function than they are like the drift diffusion model. And in fact, they are indistinguishable from a step function. So at least in our system, I can tell you that decisions are not made through gradual accumulation of information. And that brings me to the last part of the talk. Because as a psychobiologist, this only gets us so far. Because what we're really interested in is behavior. And it's not just that what I've just shown you is palatability and not behavior. So what I've just shown you is an average performance. In real life, the animal isn't determining what palatability a taste is. An animal doesn't get a taste on its tongue and say, that's sodium chloride. I like that less than sucrose, but I like it more than quinine, and so I think I'll swallow this much of it. The animal gets a taste in its mouth, and it does something much simpler than that. It says, I want to swallow this, or I want to spit it out. And so we got to really get back to that, because that's where the game is at. The animal is working one moment at a time. The animal is working in real time. And so, given what I've just shown you, we can actually go there. And so what we've done is we've gone back to our original animals and those behaviors that I showed you. Let's see if I've got that movie going. And so there you can see the gape and another gape. This movie, by the way, is very slowed down. Gapes are rhythmic. They happen about 3 to 4 a second. And we put an EMG electrode in and electrodes in the head, and now we have a simultaneous readout of when the animal actually decides to chooses to reject quinine and what the neurons are doing in that single trial. And I should say that we stuck with gaping rather than looking at both behaviors, simply because it's a lot easier to see a gape than a lateral tongue protrusion. It's a lot easier to tell that from everything else. And so we decided to stick with the thing that we could get a very, very good record of. But everything that I tell you about gapes is also true for the other decision. So this is a single trial from a new data set, and this is not Sadaka. Yes, this is still Brian Sadaka under review, but this is work primarily of my postdoc Jen Lee and my graduate student Narendra Mukherjee. And so this is the hidden Markov model solution for a single trial of quinine in this data set. And the dashed green line is the state that we've already identified as being palatability related. And that's when we could say it comes on. Those are the gapes. Here's the next 3 trials. That state comes on earlier, the gapes occur earlier. That state comes on earlier, the gapes occur earlier. That state comes on later, the gapes occur later. Remember that this is primary sensory cortex. Remember also that these are 6 to 12 neurons at a time. This is not a data set of 150 neurons. And yet, when we go across all of the trials within that entire session, one second, and we plot the time of neural transition against the time of the behavior, this being the unity line, being the two occurring at the same time, you can see that with 4 lonely exceptions of these 30 trials, the behavior always happens just after the neural transition, and because it becomes important later, this actual timing of the behavioral latency is essentially all but a uniform distribution, but when you condition it on the time of neural transition, you get an R squared of 0.662. And so that is R equals 0.662. Actually, I should tell you that this was before all of the data were analyzed. It's gone down since then. Now the R squared is only about 0.58. And so this is all of the data. These are all of the data. These are data from 11 animals. And what we've got in the x-axis is the difference between the time of state transition and the time of behavior. Positive numbers are neural leading behavior. And this is the number of trials. And what you can see, again, remember that the timing of the onset of gapes themselves is uniform. When you condition that upon the neural transition, that's not uniform. That is a strong peak right about 200, 300 milliseconds afterwards. So when sensory cortex in a single trial says gape, the animal gapes at a 1/5 of a second delay. And one more step. All of that is still correlation. No matter how much I try to convince you that that's a very impressive correlation, it's still correlation. Let's go to causation as much as we can. So in fact, Jen Lee has done 2 different kinds of causal tests of what I've just shown you. The first one involves manipulating behavior. What she did was she took a session in which, as all of our sessions, there are actually multiple tastes being delivered, but she cued quinine. And so across the session, the animal gets these tastes delivered to the tongue, but every time it's going to be quinine, a tone plays just before the taste is delivered. What happens when you do that? The rats are no dummies. Across the session, they figure out that you are actually letting them know that quinine is coming, and that reduces the latency to gaping by about 150 milliseconds. So across the session, the animal gapes earlier and earlier because it has a better and better idea that quinine is coming. And here's the control. When you don't put a cue on, you don't see a change across session at all. So anyway, you get this behavioral change, and then you can say, what happens to the neural activity? And the answer is that across this session, the onset of this late state, on average, decreases in latency by about 150 milliseconds. So as you do this, and you change the behavioral latency, you are also changing the neural latency so that it still proceeds. But let's get on to back to the optogenetics. If you knock out GC via virally delivered ArchT, knocking out sensory cortex, you in fact change the probability of gaping. This is what on trials that you shine the 40-watt laser. It's way too high in intensity, but in awake animals in a large region, you sort of have to do that. And you compare that to control when you have either a lower intensity or no laser at all, and you can see that you only get this change with the laser. And so basically, and this is within animal, so these are some trials in which there is laser, some trials in which there is not, and you basically see that you change the likelihood of gaping by knocking taste cortex out. I should throw in here, this is a small effect. You might ask, why is this effect so small? You can never mind that it's significant. And there are several answers to this. The methodological one is that optogenetics sucks. If I want to knock out neurons, I'm going to use mucimol. That knocks out the neurons. With this, there are all of these variables that no one's talking about that have to do with, how well your infection goes, how reliable your infection is throughout the region, exactly how the laser permeates the structure that is not see-through. All sorts of things play into this, and so it is the case that optogenetics just simply doesn't work as well at knocking out these neurons as other techniques. We know because we are using optotrodes to record from these neurons while we knock them out. When Dave Krupa and I recorded while knocking out with mucimol, you got a total cessation of action potentials. When you do this, you knock it down. But the other scientific reason is that, remember the diagram I showed you of the system at the beginning. Taste cortex is only one of the regions in play here. And the CPG, the central pattern generator for this rhythmic behavior, is in the brainstem. It takes input from cortex. It also takes input from the amygdala. It also takes input from the hypothalamus. It also takes input from the BNST. We are knocking down a tiny bit of a large system. It is not surprising that the effect would be small. What is surprising is that there's an effect at all. Because by classic belief, this is a sensory structure. It should not be directly involved in driving the motor behavior. And so that's all the data I have for you. And so I wanted to let you know who actually collected these data, since all I do is take trips to Brazil. And so these are people from my lab, past and present. And normally I point out who is involved in what study. But to be honest, with a 2-hour presentation, every single person on that list contributed to the data that I presented today. And so I would like to point out that just at Brandeis, I have a whole set of collaborators. I have collaborators in this room as well. But I would need to point out specifically Paul Miller. Paul is a self-described recovering physicist. And he is the person who is largely responsible for the fact that our math works. I can barely add. And Paul is really, he's the elf who makes this all actually happen. And of course, I would be remiss if I did not thank the NIH, NSF, and Schwartz Foundation for the dollars that they have tossed our way. And that's the whole story I have. And so, since there is a little bit of time left, I am not going to take pity on you. I'm going to toss in a couple of things, just a couple of things. I'm not going to actually necessarily state a strong opinion, but I want to wade into thinking about neural coding from a larger scale philosophical standpoint, and in full recognition that thinking in a Bayesian sense does change the things that I'm going to talk to you about right now, but a little defining is in order. And that's basically what I want to give you. Talk about representations. The word representation has come up a few times, and I want to offer a definition, because I think we can't actually decide how good a term it is, how useful it is for neuroscience, unless we nail it down, because I think that there are probably as many naive definitions of this word as there are people in this room. So what I want to suggest that we are talking about when we talk about representations in general is a typically arbitrary symbol for something else. Cat is a representation of an animal that goes meow. It doesn't look like that animal. It doesn't sound like that animal. It is an arbitrary symbol for that animal that we use as input for processing. If your loved one has a cat allergy, you yell, "Cat!" and your loved one will process that and know to move away. And so a representation is a symbol that is typically arbitrary for something else that is used as an input to processing. And again, this is just one offered definition, and I'm going to suggest that things we have to look out for on that basis. If you want to argue that that's the wrong definition, please feel free. I'm not actually married to any of this. Neural representation, therefore, or a neural code, I use those two terms interchangeably myself, therefore is the pattern of firing, which of course opens another can of worms, because as we've seen, when I say pattern of firing, I'm meaning something different from what other people are meaning. That means a particular stimulus. This pattern of firing is sucrose, equals sucrose, passed from the sensory system to the systems that then determine what you do with it. And so you have to get into the system the fact that it's sucrose, deal with it. And so that is what I think of as being neural representation and neural code, and I'm comfortable with the possibility that they are genuine, that these constructs are genuinely in use by the brain. But questions to ponder when thinking about neural coding. The definitions that I've just offered you imply modularity. The idea that there is the sensory system forming a representation, which is then passed on to the association cortex or the motor system, implies a dividing line between those two, where they have something. If you're going to pass something, you're passing it over something. You're passing it over a divide. Separation of systems. If the representation is input, it is input to something else. Therefore, this definition can run into trouble with too much distribution. If you start talking about your system as being distributed to the point where everything affects everything else, this definition of neural representation is going to have difficulty. I'm not saying it's going to be wrong, saying that you then have to think very carefully about how well it works, how much weight it can carry, how much work it can do for you in your theory. Similarly, this definition implies reliability. When you press the button A on your keyboard, a signal goes into your computer that you have pressed A. Your computer only recognizes that because it's reliable. If it was a different pattern of input every time, and here's where we get to, it depends on the distribution, but if it was a different pattern of input every time, your computer wouldn't know what to do with it and certainly wouldn't recognize it as an A. And so even when we're talking distributions, there has to be a certain amount of reliability to this response. A code for which there is no code book is useless. Therefore, this definition, again, runs into difficulty with response variability that isn't noise. If your response can change, if your response to a stimulus, the neural response to a stimulus can change when under different circumstances, the more that happens, the more trouble this definition has. It also has trouble, by the way, when two responses to two different stimuli appear identical. And so, well, I don't want to belabor the point, and again, the point is that there's no right or wrong answer here. There's no dividing line between representation works, representation doesn't work. But what we do know is that neural codes are affected by everything. The obvious ones is that when you're anesthetized, it changes the neural response to the same odor and other stimuli. Here is another situation where that's true. This is work by Murakami. This is work by Rinberg. Stimuli and other modality effects codes, as I've suggested in my talk today, and this is work of Asif Ghazanfar at Princeton. Responses in primary auditory cortex to a speech stimulus differ depending upon whether or not you can see the mouth of the speaker and whether that mouth is doing what you expect. So that changes the response. Spontaneous within-session changes in arousal attention affect sensory responses. This is work of my beloved ex-postdoc Alfredo Fontanini, who found that when the animal stops paying attention, it changes the responses to the stimuli, but notice that it changes them, by the way, in very particular ways. It turns out that these changes of attention, which I can talk to you about the evolutionary value of this, is a palatability-related change perceptually. We know that because even when the animal is not paying attention to the stimulus, we can still give it to him. But the take-home message is that even within a session, you massively change the response with attention. This should be no surprise to anyone. And finally, I want to mention that in addition to the stuff I told you today, we see trial-to-trial changes in sensory responses that have to do with just where the system is at when the particular time the stimulus comes in. This is work of Jean-Marc Falloux, but there's also wonderful work by Ariely on visual cortex showing that from trial to trial, the response to a visual stimulus changes depending on what the prior state of the cortex was. Joseph Fisher has also published work on this in visual cortex. And most frightening about this is that the work from Ariely shows that what look like beautiful visual responses to orientated bars occur spontaneously as the animal is just sitting there with its thumb in its ear. And so this is the situation where there are times when the system provides the response that we have said represents a particular stimulus, even though that stimulus is nowhere around. And so you either have to say this animal is right now hallucinating that bar, or you have to explain how representation can survive that challenge. And in this case, I love these data, Jean-Marc's data showing these are responses, repeated responses to a particular, I think it was an auditory stimulus. And so he pulled apart little time periods where the response is variable. And he shows that the response really isn't as variable as you think. In fact, it turns out that when you reorder the trials, from trial to trial, there might be 3 different responses that the cortex makes to the stimulus, all just depending on where it happens to be at the time. And so those are things that, again, I am not taking a stance on whether representation is a good term to use in our theories, but given the definition that I laid out, there are problems that it runs into, to some degree or another, in that these responses are variable in a way that is not noise, and these responses are basically, and these responses, I can't remember what the first thing I gave you was. These responses really disobey modular boundaries that we artificially put in the brain, such as the sensory motor boundary. And therefore, in our system, I am not comfortable calling my responses representations of the taste, when in fact they're representations of the movement from the very beginning. And that's all I've got. Thank you. I'm happy to answer any questions. So the question is whether we've tried to manipulate the transition times. And oh, God, yes. That's all we're about right now. And we're trying to do it in a number of ways. One is neurally. We're trying to actually perturb the system in and out of states, mostly out of states. But this turns out to be, among other things, a computational hassle because the HMM is an offline hill climbing procedure, and identifying state transitions online so that we can actually do a good controlled job of orientating our perturbations to that turns out to be hard. When we do complex stimuli, we see something that at first confused us, but then of course makes perfect sense. We actually delivered mixture stimuli, and so stimuli that are a mixture of two basic tastes that taste relatively complex, and what we found is it had absolutely no impact on anything that I told you. It did have an impact. The earlier activity, the activity in what we call the identification epoch, changes. It is no longer a beautiful representation of the stimuli. It isn't a beautiful linear representation of the mixture. It's something else entirely. But that transition into palatability occurs right on schedule, which makes sense. Part of the problem with comparing this work to other decision-making work is that there's no such thing as a hard version of this task, at least normally. Let's say I deliver really low-intensity stimuli. When something very bland, that doesn't make it harder to decide whether you want to spit it out or swallow it. Some people like bland food, and they're like, yeah, good. Some people don't like bland food. They're like, that sucks. But it's no harder for them to decide. Yes, and so we're getting into that now. We're getting into food in which the quality of the stimulus actually changes through time. Don't have those data for you yet, but we're working on that. But it also turns out that there is a natural way to influence the state transition time, something that I finessed in the talk because we don't know how to explain it. Turns out that the more aversive the taste is, the faster the system goes through the sequence. And so it is simply the case that it gets to that final state a little slower with sucrose than it does with quinine. And when you make sucrose aversive, it gets faster. This innocuous sounding result is pissing us off because it means that while we talk about the earlier response as having no information about palatability, that cannot be right. Because if it was the case that that earlier part of the response was palatability free, then there would be no way for the system to know to switch early. So we don't know yet what that means, but that's one of the things we're looking into, and I'm happy to have anyone who wants to come join us look into as well. So the question is, and believe me, I have heard this question. How do we know that there isn't an accumulation, a ramp occurring somewhere else in the brain, and that we are looking essentially at the output of that? And the answer is we don't know. I will say very defensively that no one has ever asked Mike Shadlin to check whether every other brain area is also ramping. And so to some extent, our response is, why should we have to look when they don't have to look? It could be that the situation is reversed. Having said that, we've looked in an awful lot of places. All of the places we've looked, including the parabrachial nucleus in the pons, which is the second relay. It comes from the tongue to the medulla, and from the medulla to the pons. We see the same kinds of transitions there. And so we've looked at a range of areas. We haven't seen it yet. Question is, a question that by all rights shouldn't be asked for another 12 hours, is what would I study instead of taste if I had the chance now? Wow, it's a little bit like asking, what college would you have gone to if you hadn't gone to the one you went to? It's hard to think. So the first question is, would I study taste again if I had the chance? And there are substantial upsides and downsides to studying taste. One of the upsides is I am never scooped because nobody else cares. It's weird. Everyone cares about how things taste. Everyone is keyed into their sense of taste. Nobody studies it. There are precisely 2 awake in vivo people recording from taste, me and my ex-postdoc. There will soon be a 3rd because another ex-postdoc is going to open his own lab. And the downside is that, therefore, nobody really cares. If you're interested in high-profile publishing, more power to you. We are shut out of that for the simple reason that those journals are businesses that rise and fall on impact factor. Every single taste researcher could cite my papers, and that would be 5 citations. And so there is no way that work like mine is going to be high profile. Would I do it again? I probably would, because I think it's easier to say the scandalous and outlandish things that I say in this relative vacuum than it would be if, God forbid, I tried to do it in vision. In fact, that's why I think whether it turns out to have been right or wrong, that beautiful paper by Sugassi in '99, which suggested that face responses are not representations of faces, but are processing faces, has never been replicated or dealt with, because whether it's right or wrong, it's just not going to get play. And so, I think I'd stick with it. In fact, I truly believe that eventually the sorts of things that I'm showing you here, if they hold up over time, will hopefully change the way other sensory systems are studied. Okay. Right. If I couldn't do gustatory cortex, I honestly think that I would be a motor researcher. I think that I would be getting closer to the output. I can't tell you how relieved I am to finally be doing work in which the other end of the neural arc is included, because it actually is like, oh, this actually is related to the animal's behavior. And so probably if I had to change, I would follow someone like Krishna Shenoy and do work at the other end of the system, just not in monkeys. Yes, so 2-part question. The first part is, why not say olfaction? And the answer is that as far as I'm concerned, olfaction has all of the problems of taste and none of the advantages. It is a difficult... That's right. It seems like it is, but it isn't, because you have to depend upon the animal sniffing. If the animal doesn't breathe in, you do not get delivery. The cannula of the mouth has huge issues, absolutely. But the issues in taste and olfaction end up being the same, in that olfaction only works when the animal sniffs, and in fact, we've been trying to find a way to take the social transmission experiment into electrophysiology. And the hard part is knowing when the smell hits the nose, because you have to rely upon knowing exactly when the animal breathes in, which we can do, but also knowing when the other animal breathes out, which we have trouble doing. It turns out to be a mess. But and in the meantime, you enter a field that's very crowded. I'm a businessman. I don't like the olfactory field very much. I think that it's kind of full of, there's jerks there. But the other part of the question was, what was the other part of the question? Yes. What happens with the coupling between olfaction and taste? You actually all really know the answer, because you've all had colds. Even down here in the warm paradise, you suffer from colds. Your nose is blocked for about a week, and things taste awful. When you close off the olfactory system, the taste system suffers massively. And that has led to a conclusion that, well, taste basically is just smell. I can't tell you how many times I've gone out to dinner and my companion has said, oh, you study taste? Isn't that just smell? And the reason that it's quite reasonable to reach that conclusion is because you can't do the natural converse experiment. You never find yourself in a situation where your mouth is blocked for a week, although we all know people that we wish that would happen to, and you try to smell things during that time. And it turns out that while it's not purely symmetric, taste and smell are mutually interactive. And so things don't taste right when the olfactory system is working, and things don't smell right when the taste system isn't working. I'd probably go further and say things don't look the same when the taste system isn't activated. Certainly, it's known that they don't taste the same when you can't see. There are wonderful experiments showing that things taste the way they look, such that the candy companies have figured this out, and all of those wonderful fruit candies that you like so much, there's only one taste in those candies. The red, yellow, green candies, they're all the same thing, but by giving you a different color, they convince you that they're different flavors. So other than what I just showed you, there is data that I did not present. So those gapes are rhythmic. And we looked for evidence that there was spiking in cortex that drove each individual gape. It's not there. And so I ran in Panic to one of my mentors, Eve Marder. If you guys are not familiar with Eve Marder's work, then shame on you, because she is one of the best neuroscientists in terms of the way she incorporates theory and math into what she does, and what she finds is revolutionary. And what she and I talk about is the fact that in her system, there is this beautiful central pattern generator. She looks at the crab and lobster stomatogastric ganglion, which is very similar to what we're looking at. It's the set of neurons that drives the rhythmic behavior of chewing and swallowing, which happens in the gut of those beasts. And what she sees is that it's multifunctional. It can produce multiple rhythms. It can produce these rhythms when dissociated from the rest of the nervous system. But in situ, there are a bunch of descending modulatory inputs. And by the way, if I could do one thing in the world, it would be to rescue the word modulatory from its second-rate status. Modulation is what goes on in the brain. Things modulate other things. And this modulatory input isn't involved in driving each cycle of the rhythm. The CPG has neurons that are half-center oscillators. It does it all by itself. But they help determine what rhythm should be produced at that particular time. And so what we think is going on in taste cortex is analogous to that. It is not actually involved in the driving of individual behaviors per se, but it is one of the regions that modulates the central pattern generator, the multifunctional central pattern generator in the brainstem, and says, gape now, please. And that's what we think is going on. And so, put in that way, and in the context of your question, perhaps I'm being too strong, purposefully. It bypasses motor cortex. Yes, this does not involve any other part of cortex, probably. Although I have a student who disagrees with me who's doing experiments on that now. But saying that there's no dividing line, it isn't the case that what's going on in taste cortex is pure behavioral driving. It is, in fact, something at a slightly higher level. It is involved in tripping a rhythmic CPG into one of the possible rhythms that it produces. Thank you very much.